So hello students, welcome to CBSE NCERT Solutions by Vedantu. On this channel, we do NCERT line by line of every single chapter from in your NEET syllabus. Now students, this is a very useful session for all the people who are aspiring for boards as well as NEET, because in biology, your entire question format will be from your NCERT line by line also. So pay very close attention to every single line of NCERT when we are teaching. Any doubt you can always ask me, and let's start the class, students. Now if I ask you a question, all of you, if I ask you a question, imagine if you could look at a hand on the inside. It'd be really cool, right? To look at how a hand looks from the inside. Or if you can scan someone's brain from just by looking at it, the internal structure of a brain. Now, unfortunately, it is very difficult to do that in the case of humans or animals, but you can easily do that in the case of plants, right? You can take a plant on the roadside, take a leaf, stem, or root, cut it open, look under the microscope, and you can find the internal structure of a plant. That is, you can find the internal structure of a plant. It's really cool, right? Now, students, once you look inside a plant, this is what will be visible. You can see the different tissues on the inside. You can see the internal lining of the inside. You can see the arrangement of vascular bundles on the inside, and it is completely legal. It is completely legal. No one will put you in jail for this, right? So that is the beauty of looking at inside of a plant. Now, in the previous chapter, that is morphology, we learned how a plant looks from the outside, that is the external features, right? In the previous chapter, we learned about the external features, that is morphology. Then what is anatomy then? Anatomy is nothing but the internal structure. Internal structure and arrangement of the cells and the tissue is called as anatomy. There's a very clear difference, students. Understand the difference. Morphology deals with the external body. Anatomy deals with the internal body. And let me tell you, you might find a flower or a root or a stem looking beautiful from the outside, but a plant looks even more beautiful from the inside. Here is a clear example of that. Hello, Mayank. Like, a plant looks very beautiful from the inside. The arrangement, the intricate design inside is even far more beautiful. So students, what is anatomy? Anatomy is a branch of biology that deals with the internal structure and arrangement of cells and tissues. Okay, any doubt, quickly ask me, students. No doubt. Now students, if you have passed your class 7th, 8th, 9th, 10th, you should be able to tell me what is the meaning of a tissue. Can anyone tell me what is the meaning of a tissue? Anyone? What is the meaning of a tissue? Very simple, right? What is tissue? Tissue is nothing but a group of cell. It is called as group of cells. Okay, it is called as group of cells. Now, are these group of cells, do they have a common origin? Do they have a common origin? I'm like, yes, they have a common origin. This group of cells, do they have a common function? We'll be like, yes, sir, they will have a common function. Group of cells, tissue, do they have a common origin? Yes. Common function, yes, but do they have a common structure? That is very important to understand. Now, if you're in class 9th or 10th, you will tell me, yes, sir, they have common structure also. Group of cells have common origin, common function, and common structure. You'll be like, yes to everything. But right now, you're in class 11th. You're in class 11th. You should answer very carefully now. If I ask you the question, do they have a common structure? There are two options will come in front of you. Two options. The first one is yes, they can have a common structure. The second one is obviously no, they do not have a common structure. Examples for common structure is going to be your xylem. One is common for structure is going to be your parenchyma. One second, friends. For the common structure, it is going to be parenchyma, collenchyma, as well as sclerenchyma. So all the following three different of tissues will have a common structure. Now, what about the non-common structure or dissimilar structure? The dissimilar structure is basically the xylem and phloem. Xylem and phloem. They will have dissimilar structure. So, what is a tissue? Tissue is a group of cells which can have a common origin, can have a common function. They can have a common function, common structure, or they cannot have a common structure in the case of xylem and phloem. Xylem and phloem. Any doubt till now, quickly ask me. A simple classification which will help you to distinguish. Okay. Now students, the next step is classifying the tissues. Now we are supposed to do something called as types of tissues. Types of tissues. Now there is a classification on what basis are we deciding types of tissues. See, in assembly, all of you stand in line, and when you stand in line, there is always a criteria, height-wise order. Yes or no? Tell me. You always, when you stand in a line, you always do it in a height-wise order. So here also, when you're trying to classify the tissue, there is a category or there is a classification. Right? On what basis? The basis here is ability to divide. Ability to divide. Based on the ability to divide, we have types of tissues. Types of tissues. Based on that, we have two main categories. We have two main categories of types. The first one is called as your meristematic tissue. The first one is called as what? Meristematic tissue. And the second category is called as permanent tissue. It's called as permanent tissue. Then what is the difference now? What is the difference? Your meristematic tissue, students, understand very carefully. Meristematic tissue has the ability to divide. Ability to divide. While your permanent tissues cannot divide. They cannot divide any further. They have lost the ability to divide. Permanent tissue. Your meristematic tissues are living cells, made up of living cells. These permanent tissues are they can be living or dead. Can be living or dead. Both possibilities are there in the case of permanent tissues. Clear, all of you? Clear? Permanent tissue. So this is the basic classification. Basic classification between meristematic tissue as well as the permanent tissue. Clear now, students? Now first we will understand what is the meristematic tissue, what are the different types of meristematic tissues, and what you need to understand meristematic tissue. And once we are done with meristematic tissue, primary meristem, secondary meristem, lateral meristem, apical meristem, intercalary meristem, once we are done with all that, then we will go to permanent tissue. Under permanent tissue, we will be understanding something called as your parenchyma, collenchyma, sclerenchyma, xylem, and phloem. So, are you ready for that? Tell me. Ready? And anytime if you have any doubt, quickly ask me. And because let me tell you, anatomy is a chapter which needs proper concentration. It needs proper understanding. You cannot mug it up. If you try to mug it all this up, it's going to be very difficult. Let me tell you why. The next concept I'll teach you. Okay. So the next concept is called as meristematic tissue. Right? Meristematic tissue. This we already discussed, that permanent cells having similar structure, dissimilar structure. Here we learned here, right? Similar structure, dissimilar structure. Okay. Now, everyone, look at the diagram. All of you, look at the diagram of meristematic tissue. What do you see when you look at this diagram? Apart from the cell division, do you notice that every single cell here has a large nucleus? Yes or no? Tell me. Do they have large nucleus like this? Like one round bindi on the top head. It looks like a round nucleus. So can I tell large nucleus is present? Large nucleus. Large nucleus which is central in place here. Central. Okay. Large nucleus central. Now apart from that, it's a dividing cells. These are dividing cells. So these are dividing cells. So they're living in nature. Living in nature. Yes, living in nature. Now apart from that, every single dividing cell, do they have dense protoplasm or very weak protoplasm? Tell me. If the dividing cell, the living tissue is very fresh and very dense, so can I tell they have dense protoplasm? They can have dense protoplasm. Now students, the cells here, do they have small size or large size? They're dividing cells. They're new cells, right? So they are small size cells. Small size. Small size cells. Okay. Now, do you see any form of intercellular space anywhere, students? Tell me. Do you see any form of intercellular space? See, aren't they compactly arranged? Compactly arranged they are. I cannot see any form of intercellular space. So can I tell inter, intercellular space, ICS, intercellular space is completely absent here. Yes or no? Tell me. Intercellular space is completely absent here. Now students, these are new cells. So they have something called as thin walled. They're thin walled completely. Thin walled cells. Thin walled cells. They mainly have the primary cell wall. Primary cell wall. Thin walled cells, right? New cells. Thin walled cells. Okay. And also vacuoles are absent here. Okay. 1 second. Vacuoles are completely absent here. Okay. Vacuoles are completely absent. So, large nucleus, living, dense protoplasm, small size, interstellar space is absent, thin walled, vacuoles are absent. After looking at the diagram, you can tell me all the following points easily, right? All the following points can be told very easily. Now, thin walled, large nucleus, no interstellar space, dense cytoplasm. Okay. Now, students understand that was your meristema tissue. That is meristema tissue, right? Now, this meristema tissue can generally be also classified into two different categories. The entire meristema tissue can be classified into two different categories based on two factors. That is, meristema tissues can be classified based on the origin, based on the origin, and based on the location. Based on the location. Ready? Now, meristema tissue based on origin. First, we'll understand based on origin. Based on origin, we have two main categories. One is called as primary meristem. Primary meristem. The other one is called as secondary meristem. Secondary meristem. Now, what is the difference between primary and secondary meristem? Understand. Very simple. Primary. Now, in the school, do you have primary first or secondary first? Tell me all of you. Do you have primary class, then you go to secondary, then you go to college, and then you go to pre-university and university, right? So primary is first class to fifth class. Yes. So primary will come in your life early stage or later in your life stage. Tell me. So primary will usually come in your early life. Same. In the case of your plant's life cycle also, primary meristem is found from the beginning. It is found from the beginning. Okay, it is found from the beginning. It is found from the beginning here in plant's life cycle from the beginning. Okay, found from the beginning. Now, it is if it is found from the beginning, what could be the use of it? Initially, you'll grow in height, right? A lot. So this plant also primary meristem is responsible for the increase in increase in primary growth. Increase in primary growth. What is primary growth? Primary growth mainly involves height increase. Height increase. That is called as your primary growth. And this primary meristem can be found in both monocot and dicot. Tell me any doubt till now. Primary meristem. Primary meristem is found from the beginning, responsible for the primary growth, found in both monocot and dicot. Very simple to understand. Very simple to understand. Now, what about secondary meristem? Secondary college is later in your later in your life. So, in the case of plants life cycle also, secondary meristem is later formed. It is later formed in plants life. In plants life. Later found in plants life. Not from the beginning. It is not present from the beginning. Now, what is it responsible for? It is responsible for the secondary growth. Secondary growth. What is secondary growth? Secondary growth is basically increase in width. Increase in width or girth. Or girth. See, in primary growth, the plant is growing in height. Nicely, it is growing in height. But in secondary growth, the width is increasing. The width is increasing. The plant will grow in width. That is called as secondary growth. Secondary growth. And the secondary meristem is only found in the case of dicots. Only found in the case of dicots. So primary meristem, secondary meristem. Classification based on origin. Based on origin, two separate categories. Okay. Found from the beginning, increase in primary growth, monocot and dicot. Later found in plant's life cycle. Secondary growth, that is width or girth, found only in the case of dicots. Only in the case of dicots. Only in the case of dicots. Clear? Clear all of you? Yes or no? Tell me. Now, if that is clear, that was based on origin. That was based on origin. Now the second classification is based on the location. Based on the location. Based on the location, we have three separate categories. Not one or two, we have three separate categories. The first one is called as apical. First one is called as apical. Second one is called as intercalary meristem. And the third one is called as lateral meristem. Lateral meristem. Lateral meristem. Clear? Now first we'll study about apical. First we'll study the apical meristem. What is apical meristem and where is it found? Apical means what? Apical means apex. This is apex. For example, this is a plant's body. I'm a plant. Imagine I'm a plant. On the top of the plant body, you'll find the apical meristem. I will tell you an example. Understand? So where is it present? First of all, it is present at apex. Present at apex. Okay. It is also responsible for the axillary bud formation. Axillary bud formation. Okay. Now it is present at top. I told you top or apex. Now, for example, if this is a seed, if this is soil. Okay. Here we have root. Root is growing in the soil. Okay. And on the top we have the shoot. We have the shoot growing on the top. We have shoot on the top and root on the bottom. Now this shoot is there, right? Can you see this tip? The tip of the shoot. This one. This right here. This tip right here is called as shoot apical meristem. See, apical, right? Apical meristem. Apical meristem. Intercalary meristem as well as lateral meristem. This is called a shoot, right? This is shoot. So this is called a shoot apical meristem. This is root apex or root top, called as root apical meristem. Both are a type of apical meristem. One is found at the shoot top, one is found at the root top. So one is called as shoot apical meristem, other one is called as root apical meristem. So in this apex, one at the shoot top, one at the root top, we find the apical meristem. So all of you know where apical meristem is found now. It is found at the shoot apex or root apex. Now students, remember this apical meristem is also increase in height. It will also help in increase in the height. So if it is also increase in height, can I also call apical meristem a type of primary meristem? Now tell me yes or no. Can I call apical meristem as a type of primary meristem? Why? Because it's present in the plant's life cycle from the starting itself. So it's also called as a primary meristem. That is apical meristem. Done. Now that was the apical meristem. Very simple to understand. Very simple to understand the apical meristem. Done. Now what is intercalary meristem and where do you find it? Intercalary meristem is found between. It is found between mature tissues. Mature tissues. What is it responsible for? It is responsible for the increase in the internode elongation. Basically, internode elongation is happened. Where do you see this? In the case of mostly in the case of grasses. In the case of grasses. So every single time a cow is coming and eating the grass, the top grass is removed by the cow. So you within few days you can see the grass is growing very fast. Why? The grass is able to grow very fast because of intercalary meristem. Because of intercalary meristem, the grass is able to grow very fast. Okay, and apical meristem is found both in the case of monocot as well as in the case of dicots. This is also found in the case of both. Mostly monocots actually. Mostly monocots. Now what is lateral meristem? What is lateral meristem, friends? What is lateral meristem? First of all, tell me where is it present? It is present on the sides. It is present on the sides. What is the meaning of that? Present on the sides. Very simple. See this. This is called as, this here we have apical meristem. Here we have intercalary meristem at the nodal region. And we have lateral meristem present on the sides. Can you see? This side and this side. Present on the sides. That is lateral meristem. Okay. That is called as your lateral meristem. Now, what does lateral meristem produce? Lateral meristem is responsible for the secondary growth. Only found in the case of dicots. Only found in the case of dicots, that is meristem, lateral meristem. So based on location, we have apical, intercalary, and lateral meristem. Three different types of meristematic tissues. Done. Done. Yes. Now students, I'll ask you a simple question now. This is a question which I ask all my students, and there is a very high 50% chance that you might not answer this. Tell me, everyone in the chat, does the primary meristem give rise to secondary meristem? Yes or no? Tell me, all of you. I'll give you 1 minute time. Think and tell me. Does the primary meristem give rise to secondary meristem? Think. All of you think. Think. Does the primary meristem give rise to secondary meristem? Just tell me yes or no. Just tell me yes or no. The answer is what? The answer is obviously no. A primary meristem will never give rise to secondary meristem directly. Never. It will never go from primary to secondary. It will never go. Then what actually happens then? What actually happens? Listen very carefully. Always remember, a primary meristem is responsible for primary growth, and it will always produce primary tissue. Always produce primary tissues. So the flow is very simple. Primary meristem to primary growth. Primary growth is because of primary tissue. Okay. So always remember, primary meristem will give rise to primary tissues, primary permanent tissues. This primary permanent tissue, students, this primary permanent tissue will again regain the ability to divide. It will regain regain the ability to divide. Regain the ability to divide. This process is called as what? De-differentiation. It's called as what? De-differentiation. Now, this primary permanent tissue undergoes de-differentiation to produce secondary meristem. Primary meristem to secondary meristem is not a direct process. A primary meristem will always produce primary tissue, and this primary permanent tissue will regain the ability to divide and become secondary meristem. The secondary meristem will again give rise to the secondary tissue. This is actually called as your redifferentiation. What is happening? Redifferentiation. This secondary meristem tissue, which has the ability to divide, it is again losing its ability to divide and making secondary tissue. This first process which I told you, this is a dividing cell. This is a dividing cell. Dividing cell is losing the ability to divide and making permanent tissue. The first process right here is called as your differentiation. It is called as what? Differentiation. So differentiation means what? Losing the ability to divide. De-differentiation means what? Regaining the ability to divide. And again, redifferentiation means what? Again losing. So first you lose, you gain, you lose again. Nice, no? Nice cycle, no? So first you lose, you gain, again lose. So a primary meristem tissue will give you permanent tissue, differentiation. That's how we get parenchyma, collenchyma, sclerenchyma, all that. Yes, that's how you get it. This primary permanent tissue will again regain the ability to divide and form secondary meristem. In secondary meristem, you need to remember about vascular cambium. You learn about it. You learn about cork cambium. All of these are responsible for secondary growth. Again, redifferentiation. Very simple to understand. Yes or no? Tell me. So, a primary meristem will always produce primary tissue. This primary tissue will develop into secondary meristem eventually. Not direct, never direct. I hope that is clear to all of you. Now let's decode this diagram of NCERT. Very simple diagram of NCERT. Let's decode it. Okay. Now here, what do you see here? Here we see first we'll do this diagram. Do you see here root apical meristem right here? So right here we have the RAM, Ram and Sham. Here we have Ram. Okay, here we have Ram. This Ram will divide on this side. Ram will also divide on the other side. When it is dividing on the bottom side, it is forming initials of root cap. This initials of root cap will develop into root cap eventually. Develop into root cap eventually. Done. Now on the top, do you see initials? Initials means what? Precursors. Before the actual product, we have the demo piece. This is a demo piece. Okay. Initials of central cylinder and cortex. We also have the protoderm here. Protoderm is basically the outer layer. And in the case of root, the outer layer is called as epidermis. The other name of epidermis is also called as epiblema. So this protoderm here, this protoderm right here is the epidermis. It is the epidermis. Also called as epiblema. That I'll teach you later on, don't worry. Epiblema. Now, initials of central cylinder. Initials of central cylinder will make up the central cylinder later on, which is basically the vascular bundles. Because obviously, outer it is epidermis, then cortex, and the middle vascular bundles. That is how it goes. Then we also have cortex. All of you know general cortex. Okay. So initials of central cylinder and cortex will make up the central cylinder and cortex eventually. Done. Done. Yes. Now we will see this diagram. Here we have something called as your shoot apical meristem. Yes or no? Shoot apical meristem. SAM. The SAM will divide on both the sides, this side and that side. On the bottom side, it will make the differentiating vascular tissues, normal xylem and phloem. On the top, it will make something called as leaf primordia. Now, if you remember from the previous chapter, that is morphology of flowering plants, did we learn that as the shoot apical meristem is growing, on top, the leaves are produced in an acropetal succession. Yes or no? See, if this is the SAM, as the SAM shoot apical meristem is growing, the leaves are produced in a acropetal succession. Leaves are produced. So can you see this leaf primordia is nothing but the leaves will open up. Can you see this hand shape? This leaf will open up eventually and produce a leaf. Now as the leaf is going on top, as the leaves are being produced, in the leaf axis some cells are left behind. Right? For example, see here students, this is the branch, this is the stem. At the nodal region we have a leaf here, and leaf, this is called as leaf axil. In this leaf axil, some cells are left behind this leaf axil. These cells will end up making the axillary bud eventually. Can you see here? Axillary bud in the leaf axil. Here, right here in the leaf axil. Now, when you read the lines of NCERT, it becomes very clear. Understand now NCERT lines. See, growth in plant is largely restricted to a specialized regions called as meristems. Yes. Small region meristem, that's where growth happens. Yes. Now, the meristem which occurs at the tip of the root, I told you, at your Ram and Sham, okay, and shoot produces primary tissue called as apical meristem. Did we learn that here? SAM, RAM, apical meristem. Done. Next. Root apical meristem occupies the tip of the root while the shoot apical meristem occupies the distinct most region of the stem axis. Okay. Now this understand this line. During the formation of leaf, as the shoot apical meristem is growing, during the formation of leaf, elongation of stem, some cells are left behind. From the shoot apical meristem, from SAM, and constitute the axillary bud. Right here. You see here? Axillary bud. Some cells are left behind the leaf axil. They make up the axillary bud eventually. Okay. Now, such buds are present in axis of the leaf and capable of growing into a branch or a flower. So students understand this particular structure right here, axillary bud, can develop into a branch, branch, or it can develop into a flower. Any of the two possibilities is possible. Okay, it is possible. Now, next one is the meristem which is found between mature tissues. The meristem which is found in the between mature tissues. See here, it is found between mature tissues. That is called as your intercalary meristem. Did we learn that here? Yes, we learned that here. Called as intercalary meristem. It occurs in grasses and regenerates parts removed by the grazing herbivores. So if herbivores is coming and eating the grass, that grass is immediately replaced by the intercalary meristem. When I say immediately, 2 3 days, 2 to 3 days. Both apical meristem and intercalary meristem are primary meristem. Both are primary meristem. Why? Because they're present in plants' life cycle from the starting stage. See here. Both this is also primary meristem. Intercalary meristem is also a type of primary meristem. While your lateral meristem is a type of secondary meristem. Secondary meristem. Responsible for secondary growth. Okay. Because they appear early in the life of the plant and contribute to the formation of primary plant body. What is primary plant body? Increase in height. Increase in height. That is called as primary plant body. Done. Done. Now, meristem occurs in mature regions of the root and shoot of many plants, particularly those that produces woody axis and appear later than the primary meristem is called as secondary meristem or lateral meristem, found later in the plant's life. Now here you need to understand, students, what is the difference? Understand the difference here. Here we have something called as fascicular vascular cambium. We have something called as fascicular vascular cambium. Then we also have the interfascicular cambium. Interfascicular cambium. Then we also have a cork cambium. Three different types of cambium. Cork cambium. Now, out of these three different cambiums, your fascicular vascular cambium is also called as your intra. It is also called as what? Intrafascicular cambium. Intra. It's present within. One second, students. It's also called as what? Intra. Intra fascicular cambium. Now the question is, where are they found and how are they found? Okay, understand this very important line. Your NCERT is making a mistake, a small grammatical mistake here. It says secondary meristem and lateral meristem are same to same, but they are not. Out of these three, all three are lateral meristem. All three are lateral meristem, but not all three are secondary meristem. What do I mean by that? Your interfascicular cambium is primary meristem. Inter is secondary meristem and cork cambium is secondary meristem. So all the three may be lateral, but not all three are secondary in origin. Secondary in origin. Okay. Hello, hello, hello. That is very important to understand. Okay. So all the three maybe they are or not maybe they are lateral meristem, but they are not secondary, all three of them. Now, where do you find them in the plant? For example, if you take a section of the root, okay, this is a small section. Now here we will do vascular bundles. Here are vascular bundles. Okay. Now, understand the diagram very well. Here we have something called as between them. Okay. Now here we have something in the middle also. I believe all of you are able to see this. Right? The red and blue. The red color one here is called as inter. It's called as your interfascicular cambium. Let's put that here. Interfascicular cambium. And this blue one, do you see the blue one? The blue one is called as intrafascicular cambium. Intrafascicular cambium. Now, what is the difference? Inter is secondary in origin. Why? Because in this spot right here, before we had something called as medullary race. Medullary race. The medullary race got converted to interfissular cambium, secondary in origin. Medullary race was a primary permanent tissue. Was a primary permanent tissue. The primary permanent tissue regained the ability to divide. It underwent dedifferentiation to produce interfissular cambium, second in origin. While the interfissular cambium is primary in origin. It is primary. Nothing was there before. It did not undergo dedifferentiation, just differentiation. Okay? The difference should be clear to all of you. Simple difference. Now, there are examples of lateral meristem responsible for secondary growth. All three are examples of lateral meristem. Okay. Now, what is permanent tissues or mature cells? Permanent to the mature cells have a fixed function. I told in the starting, meristematic cells, permanent cells. Okay. They lost the ability to divide and primary tissue. During the formation of primary body, specific regions of this apical meristem produces dermal tissues, ground tissue, and basal tissue are produced. Here we learned intercalary meristem is of three types: intra, inter, and core cambium. Three different types of cambium under lateral meristem. Okay. Till here, any doubt quickly ask students. Till here, any doubt quickly ask me, students. Any doubt till here? Any one of you? Any one of you? Any doubt till now under your meristem, because meristem is over now. Now we will go to something called as permanent tissues. Ready? Ready, all of you? Permanent tissues now. Ready? Now, what is permanent tissue, students? Permanent tissue can be of two types. First of all, they have lost the ability to divide. Permanent tissues have lost the ability to divide, and they can be further classified into two categories. The first one is called as simple permanent. Simple permanent. Other one is called as what? Complex permanent. Complex permanent. What is simple permanent, friends? Simple permanent has only one type of cell. Only one type of cell. They only have one type of cell. Complex tissue, they'll have more than one type of cell. More than one type of cell. Okay, only one type of cell. Here example, all of you know, example of simple permanent is parenchyma, collenchyma, and sclerenchyma, as well as sclerenchyma. But in the case of complex permanent, we have something called as your xylem and phloem. Xylem and phloem. Done. Done. Xylem and phloem under permanent tissue. That is complex permanent. Because they have more than one type of cell. Xylem fiber, xylem parenchyma, xylem tracheids, vessels, right? So different types are there. Companion cells, sieve tube cells, everything is there when it comes to complex permanent. So, simple permanent, we have parenchyma, collenchyma, and sclerenchyma. All three are present in the case of simple permanent tissues. Okay. Now, first we understand parenchyma. Very simple, easy to understand. I will teach you line by line, and I'll make you write down some information. Write down the information. If you want all of these notes, it is already available. All of these notes are already available on the Telegram channel. This is my Telegram. Basraj Sir Biology. You can join the Telegram channel and get all the notes there. Okay, because these notes I will help you in for when you run to quick revision and everything. These notes will help you, or you can also make your own notes from these notes. Okay, now first understand parenchyma. First of all, it is the most common tissue. It is a living tissue. Living tissue. It is a living tissue. Okay, it is a major component of all Plants organ. You could take any organ. It could be stem, it could be leaf, it could be your branches. Parenchyma will be there. Parenchyma will be there. And the main function of parenchyma is actually packaging. You might think parenchyma help in photosynthesis more. No. The main function of your parenchyma is package. Every single place you find some free space, pack it. Pack it nicely. That is the function of parenchyma. Okay. So what is the main function? Main function is packaging. Package. Main function is package. The shape, can you see the shape here? The shape is almost isodiametric. What does the meaning of iso? Iso means same, diametric means diameter. So they have same diameter throughout here. Same diameter throughout. Okay, same diameter. Isodiametric. Apart from that, there is also something called as a rope. Ropes. What is ropes? It can be round, it can be oval, it can be your polygonal, elongated, and spherical. Round, spherical, polygonal, elongated, and spherical. Any of the following shape can also be present in the case of your parenchyma. Very simple to understand. Okay, all three are very good. Sound is very no, sound is very proper only, no. All three are very good. No, sound is proper. What are you saying low and all? I just now saw the sound is proper. What is this? Increase the volume in the phone, no? Increase the volume or use earphones. It'll be easy. Okay, so it can be round, oval, polygonal, elongated, or spherical. Any of the following shapes can be present for parenchyma. Clear? Clear, all of you? Yes. Now, students, apart from that, let's look at the function. What is the function of parenchyma? What is the function of parenchyma? That's all you need to know parenchyma. Most abundant, most common shape. That's all you need to know about parenchyma. Most abundant, most shape. Okay. Apart from that, one more point I'll tell you. The interstellar space. The interstellar space. Can you see here anywhere? Tell me. Interstellar space can be less or completely absent. It can be less or completely absent here. Can you see some of them? But in many cases, interstellar space is completely absent also. Okay, very simple to understand. So 1, 2, 3, 4, 5 points. 5 to 6 points are there for parenchyma. That's all. Okay, 5 to 6 points. Clear, done? Chat, all of you. Apart from the last point, if I were to ask, it's called thin walled. Thin walled. Thin walled made up of cellulose. Cellulose. Thin wall made up of cellulose. Now, what are the points here? Parenchyma. I'll repeat again. So, if you don't understand any point, quickly ask me. Most common tissue, living tissue, living tissue. Interstellar space is completely absent or very less. Major component of all plant organs. Packaging, main function. Thin walled, shape is isodiametric or the shape can vary from round, oval, polygonal, elongated and spherical. That's all is for parenchyma. If you know this, you can write the functions also very well. Because what is, for example, students, for example, the main function apart from packaging is just storage. Storage. Keep storing. Pack and store, pack and store, pack and store. Okay. Then the second thing is going to be if a parenchyma can do photosynthesis. In such case, parenchyma is called as chlorenchyma. It is called as what? Chlorenchyma. Why? Because it is chlorophyll, so chlorenchyma. Parenchyma is becoming chlorenchyma. Okay. Then the other thing is going to be gas exchange. If a parenchyma is helping in gas exchange, that is spongy parenchyma. Spongy parenchyma helps in gas exchange. Then it can also help in buoyancy. Can help in buoyancy, that is floating on top. That is called as aerenchyma. Aerenchyma. It can also secrete some things. Also secrete things, release certain substances. That also is a function of parenchyma. That also is a function of parenchyma. Clear? Done. Students, next one is called as collenchyma. Collenchyma is it is mix of your sclerenchyma and parenchyma. See, first of all, it is living. It is living tissue. Like a parenchyma, it is living. But like collenchyma, it is also producing mechanical strength. Also giving mechanical strength. So it is a living tissue which provides mechanical strength. Amazing, no? Amazing. So that is your collenchyma. That is your collenchyma, living mechanical tissue. Living mechanical tissue is called as your collenchyma. Collenchyma. Okay. Now, in the case of collenchyma, there is properly depositions on the side. Can you see here? Depositions on the side. If you look at your NCERT diagram also, NCERT diagram, can you see your depositions? Thickened corners, can you see? Thickened corners here. Everywhere thickened corners. Yes, you can see thickened corners. Now, why the thickened corners? Thickened corners is because of pectin depositions. It is because of pectin depositions. Pectin, cellulose, hemicellulose, all the following. All the following is possible. Pectin, cellulose, hemicellulose depositions are there in the corners. That's why the corners are extremely thick word. Thick, 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 thick. Okay? Done. Now let's understand the next point. That is, where do you find it? It is found below the epidermis. Below the epidermis. Below the epidermis, what do we have? Hypodermis. So it is found where? It is found in the hypodermis. For example, this is the outermost layer that is called as epidermis. Just below the epidermis, the layer is called as what? Hypodermis. It is found in the hypodermis. Now, the very important point, listen to me very carefully. That is not found in, not found in monocots and roots of dicot. That's all. Collenchyma is completely absent in the case of your monocot and also the roots of dicot. Also the roots of dicot. Then where do you find it? It is found mainly in the case of your petiole, stem, leaf of your dicot. That's all. Of the dicot. That is very important line to understand. Now, what could be the main function here, students? What could be the main function here? What could be the main function here? The main function here is obviously photosynthesis because it is living and it has photosynthesis. It has chlorophyll. Photosynthesis. Apart from that, it also provides mechanical strength. Mechanical strength. Yes. And also elastic property. Elasticity to the plant. Especially the petiole. The petiole will not able to bend very much. That is because of collenchyma. That is because of collenchyma. Next one is called as your sclerenchyma, which is a dead tissue, completely gone case. Okay, that it is a simple dead tissue. If it is a dead tissue, obviously no protoplasm. Protoplasm is completely absent in the case of your sclerenchyma, dead tissue completely. Nothing is present there. Nothing is present there. No protoplasm. Okay. Then what would be the function? The main function is mechanical strength. Mechanical strength. The reason why plants are able to stand very tall like this, no? Can you see? That is because of sclerenchyma. If there was no sclerenchyma, plant won't be able to stand very well. There will be no strength at all. Okay, mechanical strength. Now they can also have something called as pits. Also have something called as pits. I'll tell you what is pits. We'll get into it. Okay. Now students, your sclerenchyma is very special because it can be classified further. It can be classified further based on function, origin, and structure. Based on three different things, we can classify this sclerenchyma into two separate categories that is called as fibers and sclerets. Two separate classification when it comes to sclerenchyma. What is the difference? Understand the shape. First of all, shape. Look at the fibers. The fibers, if you look at the fibers, they're elongated, narrow, tube-like structures with the tapering ends. See, thin-walled, elongated cells generally occur in groups. They can have pits. Simple or bordered pits can be present. While your sclerets are shape can vary, and here the lumen is completely filled by the lignin depositions. Which depositions? Lignin depositions. See, lignin in the case of sclerenchyma, pectin in the case of your Pectin in the case of collenchyma, and in the case of parenchyma, it is cellulose. Cellulose, pectin, and lignin depositions. Lignin depositions. Okay, so highly thickened walls, they are much more thick. Shape vary, narrow lumen, narrow cavity. Lumen is completely obliterated, meaning what? Lumen is completely filled with lignin depositions. Now here, understand what is a pit. For example, if this is a cell, outer wall is called as your cell wall, and inner wall is called as your plasma membrane, PM, PM and CW. Okay, cell wall and plasma membrane. In this, between this, in the cell wall deposition, there is nicely lignin depositions. Nice, there is lignin depositions. Can you see? Nicely, there is lignin depositions. Now, in this lignin depositions, do you notice that there are small gaps? There are small gaps here. These gaps in the cell wall where the depositions is not present, those gaps are called as pits. Pits. Simple. They are called as pits. J aspirant. If you want to understand about plants, you can watch it. For example, uh, Vijay, tell me, do you drink sapota milkshake or chiku sapota milkshake? When you drink sapota milkshake, do you get that after taste in your mouth, that fiber thing in your mouth? Have you noticed? Or if you eat the the pulp of guava, also sometimes that roughness comes in your mouth when you're eating the fruits. Chiku especially, that white thing in your mouth. What is that? That is basically the sclerets. That is basically the sclerets. So if you want to know about plants more, if you want to know about more about plants more, you need to know about sclerets. Okay. So what is proper only? What? Or you need to know about sclerets. Some scam you are sometimes. Okay. So where do you find sclerets? Sclerets is mainly found in the fruit walls of nuts, fruit pulp of guava and pear. So in these walls you will find sclerets. Seed coat of legumes and tea leaves. In such places you will find sclerets. Okay, you will find sclerets in such places. Okay. Can you see? Sclerets in the coconut. In this pear. In this pear also we find sclerets here. NCI diagram, parenchyma, collenchyma, this is your collenchyma, this is your parenchyma, and this last one is called as your sclerenchyma. Fibers and sclerets. Okay. So what is complex tissue, friends? What is complex tissue? Complex tissue is a tissue which can be found more than one type of cell. Now here we have something called as your xylem and phloem. Xylem and phloem, two different types of complex tissue. Now students, we will take a 20 minutes break now. After the break, we will finish complex tissue and we will end today's class. Okay, so we will take a break now. Class resume at what is the time now? 2:14, 2:35. 2:35 will start. Okay, 2:35 will start the class at 2:35. Okay, I'll come back. Okay, students. Now, previously finished something called your simple permanent issues, done and dusted. Parenchyma, collonchyma, sclerenchyma, all three are done, right? Now we will go to what is the complex permanent tissue, where we have something called as xylem and phloem. Each one of them have something components. So we'll try to understand each one of them and write down the few points. First, we'll talk about in xylem also something called as primary xylem and secondary xylem. Primary xylem is present in the plant's life cycle in the starting stage, like your parenchyma, collonchyma. That time it's present. Secondary xylem is produced later on. Secondary xylem, which is wood, right, is present later on. Phloem also we have primary phloem, secondary phloem, present later in the plant's life. Okay? Now, first we understand xylem, all of you know. Xylem. Can anyone tell me, xylem, does it help in conduction of food or water? Simple question, no. Xylem helps in what? It helps in conducting water. It helps in conducting water and minerals. Water and as well as minerals, both of them. Okay? Now, xylem can be living and non-living, both are present. Some of the xylem elements are living, some of the xylem elements are non-living. I'll discuss that in next slide here, right here. Okay, it can be living or non-living. Now, direction. How does the conduction of water happens? Direction. For example, see here, the water is being absorbed in the roots. The water is being absorbed in the roots. From here, the water will go on top. Can the water come from the top to bottom? No, it will never happen. Always from the bottom, where the water is present in the soil, the water is taken from the roots and it is growing upwards. Can I call it unidirectional now? Unidirectional. I can call it unidirectional. So unidirectional flow happens in the case of xylem. Now xylem has four different subcategories or four different types. That is tracheids, vessels, xylem parenchyma, and xylem fibers. Out of this, all four of them, three are non-living. Only one is living. So can anyone guess which one is living here? Tell me. Which one is living? Obviously, xylem parenchyma is living. So all the other three are non-living in nature. Tracheids, vessels, xylem fibers, all three of them are non-living. Only and only xylem parenchyma is said to be living in nature. Now we will go one by one. What is tracheids? So students, here is a small trick to remember this. This is how I used to remember. It'll help you also. Think of tracheids as the pipes which is coming to your house and where you open the tap and the water is running. So in your house we have long taps, unicellular long taps, and at the end of a tap, at the end of a pipe, do you have a tap? Yes, you have a tap like this. So can I call this tubes as, can I call this pipes? Can I call this pipe a tube-like structure? Yes, it is a tube-like structure. Elongated, yes, it is elongated. Is it ending? Yes, it is tapering, right? It's tapering end. It is tapering end, right? So is it living or non-living? Obviously non-living. What is the main function? Transport of water from this pipe. Now, same information for tracheids. Elongated tube-like cell, tapering end, thick lignified walls. That is why it is non-living, lignified walls. Inner layer of cell walls have thickening from wearing in form. Dead cells without protoplasm. Most important line, one of the main water transporting elements of xylem. That's all. Small pipe coming to your house. And this is unicellular in nature. It is going to be unicellular in nature. Okay. It is going to be unicellular in nature. Clear all of you? And it is found in the case of your gymnosperms and pteridophytes. 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 Now, what about vessels? Think of vessels as large pipes going out of your house into the drainage. Now, have you noticed all the large pipes are somehow connected to each other? They're not a single pipe. Yes, they're not a single pipe. Many pipes are joined together, right? So, can I tell your vessels are again elongated tissue itself, elongated cells itself, but they are multicellular. They are multicellular. Can you see here? Each individual one is joined to another one, and they make a long chain like this. Multicellular in nature. Now, how about the connected friends? Here, this point of attachment is open. Here they will have something called as perforated suit plate. Can you see here? It is open here. So they have something called as your interconnections. Can you see here? Like holes here. Through this hole, water can pass very easily. Through these small holes, water can pass very easily. Water can pass very easily. So think of them as a large pipe with a large lumen where water can go very easily. Okay, so long cylindrical tubes made up of many vessel members. So each one is called as what? Each one is called as a vessel member. Vessel member. Vessel member. Clear? Each one is called as a vessel member here. Each one has lignified walls, a large central cavity. That's where a lot of water has to go out of in the drainage pipe. No protoplasm. Interconnected through the perforation in the common wall. Can you see? This is the common wall. And here we have perforations. Perforations are there. Typical feature of angiosperms. So it is found in the case of angiosperms. Your tracheids is found in the case of gymnosperms and pteridophytes. That's all. Tracheids and vessels, done. Nothing else you should know about tracheids and vessels. See, comparison. Present in all vascular planes, present only in your angiosperms. Narrow lumen, wide lumen. Highly thickened, less thickened than tracheids. Do not have end plates, have end plates. Okay. Here we also have tapering ends, non-tapering ends in the case of vessels. Now, xylem parenchyma is very simple. All the parenchyma same function will be there. Living tissue, main function is storage. Main function is storage. Living tissue, thin walls, cellulose, like a parenchyma. Function is what? Storage of food in the form of starch, fat, and other substances like tannins. Storage. Xylem fibers. Fiber is non-living. All of you know. Non-living, highly thickened walls. Obliterated central lumen. What is the meaning of this? Understand. This is a tissue, right? These are fibers. Inside part is called lumen. When the lumen is completely covered, when the lumen is completely filled, that is called as obliterated. That is called as what? Obliterated. Obliterated. Now, what is the meaning of septa? Septa means cross wall formation. Sometimes what happens is there is cross wall formation in this cross wall formation. That is called as septate. This is called as aseptate. No cross wall formation is called as aseptate. Then aseptate and septate. Oh, watching Principles of Inheritance. Principles of Inheritance is a very good chapter. Soon I'll be teaching that on YT also. Principles of Inheritance I'll teach again. Probably next week. Next week we will do line by line of NCERT. Next week we will do line by line NCERT of Principles of Inheritance. Don't worry. That is coming up. Okay. So xylem fiber is done. See, very simple. Tracheids are small pipes going in your house. Vessels are larger pipes going into the drainage. Parenchyma is simple. Fibers also is very simple. All the two are very simple. Okay. So it's very simple to understand xylem, tracheids, vessels, fibers, and parenchyma. Done. Okay. Now we have phloem. Before we go to the phloem, there is something called as endarch and exarch. Understand what is the meaning of endarch. Endarch means protoxylem towards the center. Can you see? Protoxylem is towards center means what? Towards center, it means towards pith. Towards the pith. Metaxylem is on the periphery, outside here. But the best way to remember is a small method which I have, that is RX bike. RX. RX bike with Prem or Priya or whatever you want to name. Okay. So what is R? R stands for roots. X stands for exarch, and X is the person who left your life. So protoxylem, P for protoxylem is on the outside. Is on the outside. Protoxylem on the outside, exarch condition present in the case of roots. Present in the case of roots. Done. So protoxylem towards periphery, that is outside, exarch condition and present in the case of roots. Opposite in the for your endarch. Opposite for endarch. Okay, present in the case of stem. Protoxylem towards the center here. It's periphery here. That's all. So the only thing you should remember here is mainly the exarch condition in the case of roots, endarch condition in the case of stem. These two lines, if you remember, you're good to go. Okay. Now phloem. Tell me, what is the main function of phloem? The main function of phloem is transport of food. Yes. Transport of food. Yes or no? The food transportation can happen. Yes. Yes, food transportation can happen very easily. Now, apart from that, it can also, it's a type of conducting tissue. Conducting tissue. Now, direction. Understand direction. For example, this is a leaf right here. This is a branch. This is a tree. Here we have the canopy. And we have a leaf. For example, here we have a leaf. On this leaf, the food is being prepared. Some food will go on to the roots also. Some food will go to the shoot also. For example, here we have a flower. Here we have a flower. The food will go here also. So can I tell the food which is prepared in the leaf is going on the both the directions? Yes or no? So can I call it bidirectional now? Can I call it bidirectional? Bidirectional in nature? Yes or no? Bidirectional. Now under phloem also we have something called as sieve tubes, campanels. These two we will study together. They are called as sister elements, sisters. Then phloem parenchyma, phloem fibers, both are very easy. So focus on sieve tubes and companion cells. That is where many questions will come from. Okay, sieve tubes and companion cells. Now out of this all the four, now in the case of, where is that? Here we had 3:1 ratio. 3 non-living, 1 was living. Now here also in the case of phloem also we have a 3:1 ratio again. But 3 this time is living. Can anyone tell me which of the following 3 is living in nature? Tell me. Which of the following is living in nature? Living is living in nature. Sieve tube is element is living. Companion cells is living. Phloem parenchyma is living. The phloem fibers is non-living. Understood? So 3 are living and 1 is non-living right now. 1 is non-living. Ulta it became full reverse. Okay. Now students understand both of them together. Very important. What are the sieve tube elements and companion cells? First, before you understand about them, understand the structure and where are they present here. We will show you NCERT diagram for that. Can you see the NCERT diagram here? Yes. Now can you see here? First we will focus on sieve tubes. First, we focus on sieve tube elements. Can you see, sieve tube elements are long cylindrical cells. Can you see long elongated cells here in the middle? And do you see here, your sieve elements do not have any type of nucleus. Nucleus is completely absent. But if you look at companion cell, the pink color one, that is the companion cell. Companion cell is present next to the sieve tube. Yes. Do they have nucleus? Yes, they have nucleus. Sieve elements do not have nucleus. Companion cells have the nucleus. Have the nucleus. Okay. Here in the outside we have phloem parenchyma. Ignore this one. Ignore phloem parenchyma on the outside. Now can you see here? There's crosswell formation here. Pores are present here. Pores, sieve pores through which transportation of food can happen. Yes, transportation of food can happen. In the case of vessels, we had also had the sieve plate there. Yes. Okay. So crosswalls are here. Sieve pores are through which transportation can happen. Can you see here also? Pores are present here. Here also. They're called as sieve pores. Sieve pores. And that is how they can also talk to each other. They can talk to each other and transport can happen. Okay. Let's read what is given here now. Long tube-like structures. Yes. Long tube-like structures. Arranged longitudinally. Yes, they're arranged longitudinally. Longitudinally. Okay. Associated with companion cells. Yes. Sister cells. End walls are perforated in a sieve-like manner. Yes. Can you see here? Yeah. Perforated here. Perforated. Perforated in a sieve-like manner. Can you see here? Perforated. Mature sieve elements has peripheral cytoplasm, large vacuole, and lack nucleus. Understand this line. Very simple. If the transportation of the food has to happen, in the middle, do we need more space? Yes or no? Obviously, we need more space in the middle. So for that very reason, all the cytoplasm is moved towards the corner. It is a living cell. All the cytoplasm is moved towards the corner. You see, it has peripheral cytoplasm. Peripheral cytoplasm. And large vacuole for storage purposes. And lacks nucleus because nucleus would be hindrance. So nucleus is completely removed. So all the food can travel very easily now. All the food can travel very easily in the middle. Now, if it doesn't have nucleus, how is the functions controlled? If it does not have a nucleus, the sister is controlling the function. That is, function is controlled by the nucleus of the companion cell. See, first point is what? Long tube-like structures, done. Elongated, done. Arranged with companion cell, done. End walls perforation, done. So you have large vacuoles, lax nucleus, and peripheral cytoplasm, done. Functions is controlled by nucleus of companion cell, done. All the points are very easy to understand. It is only a matter of you remembering it now. Very easy to understand all the following points. Okay. Now see a companion cells. Specialized parenchyma cell, first one, done. Closely associated with the sieve tube elements. Obviously, can you see? Close association is there. The sieve tubes in companion cells are connected by pit fields present between common longitudinal walls. In the common longitudinal walls, they have pit fields. Can you see here? Pit fields here. Pit fields. They're connected. That's how they can control data, can talk to each other basically. Okay. Now, what is the main function? Main function is maintain pressure gradient in sieve tube. It is maintaining the pressure gradient in sieve tube. That is the difference between sieve tube and companion cells. Companion cell is like a supporting agent to the sieve tube. The main transportation is done by sieve tube, and companion cell is helping. Like in the biomolecules, we learn something called a holoenzyme, which is made up of apoenzyme and cofactor. Now here, this is your apoenzyme. This is a cofactor. It is together they're functioning like a holoenzyme. Understood, all of you? No, students. Parenchyma, very simple again, students. Very simple. The main point here is this one. Let's get to that. Elongated tapering cylindrical cells, dense cytoplasm nucleus, just like a parenchyma. Living cells like a parenchyma. Plasmodesmal connections are present, that is cytoplasmic connections. Storage of food, that is the main function of parenchyma. Okay. Absent in the case of monocots completely. Phloem is what? Made up of sclerenchyma because dead tissue. Present in primary phloem, absent in primary phloem, present in secondary phloem. So can I tell the phloem fiber is present later in plant's life? Later in plant's life. Why? Because it is present in secondary phloem, not in primary phloem. Elongated unbranched, pointed needle-like apices. Can you see a needle-like apices? At maturity, loses the protoplasm, becomes dead tissue. Friends, let me tell you, download these notes, and if you're aware, all of you know the notes are already been sent. The notes are already been sent on the Telegram channel. That is Baswaraj Sir Biology. On this Telegram, the notes are already being sent. So download these notes, read the few information, read the NCERT after this. No one can take away a single marks from your students. Anatomy is that easy. Anatomy is so easy. Okay. Now, clear students? Tissue system. I think we will do this in next class. Epidermal, ground tissue system, the entire division. All we will do in the next class. Next class. I think we will stop here today and download these notes, friends. Download these notes. Very important. You can understand a lot from these notes. Read NCERT just one more time after these notes, right? You will have the entire concept clear to you, right? So thank you so much, friends, for today's class. I will see you in the next class where we'll be doing the part 2 of the anatomy in the coming week. Okay. Thank you so much, students. Bye bye. Take care, all of you.